Welcome back to the deep dive. Today we're tackling something that honestly feels like it was ripped straight out of a sci-fi novel,
Oh, absolutely.
but it is actually happening right now in biotech labs. We are digging into the idea of the undruggable code.
It sounds so dramatic, doesn't it? The undruggable code. But honestly, that word undruggable, it's basically the final boss of modern medicine.
Yeah, and when I first looked at the source material for our deep dive today,
Mm-hmm.
I kind of assumed undruggable just meant we hadn't found the right medicine yet.
Right, like a needle in a haystack.
Exactly, like we're looking for this needle and we just haven't looked
Yeah.
hard enough. But reading through this report, it seems like the problem isn't the needle at all. It's the haystack itself.
That is a brilliant way to put it. Because when we say undruggable, we aren't talking about a lack of effort from scientists. We are talking about strict physics and geometry.
Mm-hmm.
We're talking about proteins, and specifically these master switches in your body that control things like cancer signaling, immune responses, neurodegeneration.
Really heavy hitters.
The heaviest. And for decades, researchers have looked at these proteins and just hit a massive brick wall.
Because of their shape. Right. I mean, I remember in high school biology learning that enzymes and proteins are like locks, and drugs are the keys. You put the key in the lock, you turn it, and boom, you cure the disease.
Precisely. That's the classic lock and key model. But for that to actually work, the protein needs to have a pocket,
Right.
a little groove, a crevice, some kind of cave where a small molecule drug can slot in and stick. But here is the major problem with these undruggable targets. When we look at them through, say, X-ray crystallography, they don't have pockets.
They're just smooth.
Smooth as a sheet of glass. I mean, imagine trying to rock climb up a highly polished marble wall.
There's nowhere to put your hand.
Exactly. There's no grip. And if you can't grip the protein, you cannot drug it.
Which is pretty terrifying when you think about it.
Yeah.
You're telling me the proteins responsible for some of our worst diseases are basically wearing Teflon armor.
That was the consensus for like 40 years.
Mm-hmm.
But, and this is really the pivot point of our entire conversation today, it turns out that whole smooth surface idea is kind of an illusion.
Wait, an illusion?
Yeah, it's a trick of the camera. When scientists take a picture of a protein structure, it's usually this static snapshot. We call it the apo state. It looks like a rigid statue.
Okay.
But inside your body, proteins aren't statues. They are vibrating, wiggling, breathing molecular machines. They're incredibly dynamic.
So they're constantly moving around.
Constantly. And what we've discovered is that as they wiggle and twist, little pockets actually do open up.
Oh, wow.
Yeah, but they're incredibly shy. We call them cryptic pockets.
Cryptic pockets. I love that. Sounds like a spell from Dungeons and Dragons.
It basically is. Because these pockets might only exist for a microsecond, or they might only open up if something specifically forces them to, which we call an induced fit.
So they're hidden when the protein is just sitting there by itself, but they appear when the protein gets excited or interacts with something.
Exactly. So the pockets are there. They're just playing this really high-stakes game of hide and seek.
But then, how do you design a drug for a pocket that you can't even see? One that might only exist for the blink of an eye.
That right there is the billion-dollar question. And up until recently, our methods for finding these little ghosts were, well, they were really blunt instruments.
Yeah, I read about the old method in the background notes. It was called mixed solvent molecular dynamics, which sounds very advanced, but when you strip away the jargon, it sounded kind of messy.
Oh, it was incredibly messy. Think of it as the flooding method. Scientists would run a computer simulation of the protein and basically just drown it in a mixture of water and organic solvents, usually benzene.
Benzene, like the stuff in gasoline.
Similar chemical family, yeah. Benzene is really sticky. It likes to find oily, hydrophobic spots. So the theory was if you flood the protein with benzene in a simulation, the benzene molecules will eventually cluster wherever those hidden pockets are.
So you're just marinating the protein in lighter fluid and hoping the fluid finds a hole.
Pretty much. But think about that for a second. If you're listening to this, is that how proteins exist in your body? Floating in a pool of benzene.
I really hope not.
Exactly. It's a totally unnatural state. So in these simulations, the benzene and the water would often separate, kind of like oil and vinegar in salad dressing.
Right.
And you'd get these massive blobs of solvent probe aggregation, we call it, that had absolutely nothing to do with the protein itself. And even worse, those harsh solvents could stress the protein so much that it would literally unfold.
Oh, so you aren't finding a pocket. You're just breaking the lock completely.
You're destroying it. And on top of that, you run into this nasty circular logic problem. It's called biased sampling.
How does that work?
Well, to run a good computer simulation to find a pocket, you usually have to tell the computer roughly where to focus its computing power. You have to define these things called collective variables. But to define those variables...
You need to know where the pocket is in the first place.
Exactly. And you
Right.
can't find it without knowing where it is, and you can't know where it is without finding it.
Wow. It's a complete catch-22.
It was, until now. Because the source material we're diving into today is a report on a brand new AI system that entirely bypasses this loop. It's called the Hierarchical Neurosymbolic Multi-Agent System, or HNS-MAS.
HNS-MAS.
Ah,
Really rolls right off the tongue.
it is a terrible name for an incredible piece of technology. Instead of using one giant AI model to just guess the shape, they've built a digital crew. It is a multi-agent system.
I really want to frame this for you listening, because this is the part that totally hooked me. It feels exactly like an Ocean's 11 heist movie.
Yes.
You don't just send one guy to rob the Bellagio. You have a whole team. You have the scout, the strategist, the safecracker, and the guy in the van checking the security feeds.
That is surprisingly accurate to how this architecture works.
Mhm.
Each AI agent in the system has a highly specialized role, and they constantly pass information back and forth to crack the protein.
Okay, so let's meet the crew. First up, we have the scout. In the paper, they call this the perceiver agent.
Right, the perceiver. Its main job is target adaptation. And to understand why this is such a big deal, you have to understand the major flaw in most biological AI out there today, like the early versions of AlphaFold.
AlphaFold is the big Google DeepMind one, right? The one that predicted basically all the protein structures.
Yes, and it's brilliant. It changed biology. But it was trained on the protein data bank, the PDB.
Which is a library of all the proteins we know.
Exactly. The problem is that library is heavily biased. It is full of nice proteins that are stable, easy to photograph, easy to study in a lab.
So it's like training a detective by only showing him pictures of jaywalkers and then asking him to go catch a serial killer.
That's a great analogy. The undruggable proteins, the ones causing these horrible diseases, are messy. They're disordered. They do not follow the neat rules of the training data. So if you use a standard model on them, it hallucinates. It tries to force the weird protein to look like a normal one.
So what does the perceiver agent do that's different?
It uses a technique called protein TT. That stands for protein test time training.
Okay, acronym alert. Break that down for us.
Think of it like this. A standard AI model learns everything before the test. It completely stops learning once you deploy it. But the perceiver agent, when it sees this new weird protein, it actually pauses.
It stops.
Yeah, it basically says, I don't recognize this dialect. And it retrains itself on the fly just for that one specific protein.
Wait, so it crams for the exam while it's taking the exam?
In a way, yeah. It minimizes its own perplexity, its internal surprise at the sequence. It fine-tunes its parameters until it deeply understands the specific grammar of that one target. It builds a completely bespoke model on the spot.
Okay, so the scout has cased the joint. It understands the terrain. But just knowing the layout isn't enough to pull off a heist. You need a plan. Enter the strategist.
The planner agent. This is layer 2, and this is where it gets psychologically really interesting. The planner uses a hierarchical reasoning model, or HRM, and it's explicitly designed to mimic the human brain.
Mimic the brain how?
Well, think about how you think. You have fast, instinctive thoughts, like catch the ball. And you have slow, deliberate reasoning, like how do I solve this algebra equation?
System 1 and System 2 thinking.
Exactly. The planner has both. It has a high-level module for big picture, slow strategy. It asks abstract questions like, where might a pocket theoretically open based on the allosteric hotspots?
And then it has a low-level module for the grunt work.
Right, for the fast geometric refinement. But the absolute coolest feature of the planner, and this is something I honestly wish more humans had, is that it has self-doubt.
Self-doubt. You're telling me they built a robot with anxiety.
A robot with humility. It uses a feedback loop called CLIO. Cognitive loop via in situ optimization. See, in traditional AI, if the model doesn't know the answer, it usually just guesses. It hallucinates a confident solution because it's programmed to always give an answer.
Fake it till you make it.
Right. But the planner agent constantly monitors its own confidence. If it gets confused about where a pocket might be, CLIO kicks in. It enters a belief state.
A belief state, meaning what?
Meaning, it effectively stops and says, I am not confident in this path. I need to rethink my strategy.
So it doesn't just plow forward into a mistake. It actually backtracks.
It adapts, and it builds a belief state graph so the human researchers can actually see exactly why it changed its mind. It shows its work step by step.
Which is crucial for scientists. Okay, so we have the map from the scout, and we have the plan from the strategist. Now we need the safe cracker, the one that actually opens the vault door. This is layer 3, the execution agent.
The muscle of the operation. This agent runs on the Isomorphic Labs drug design engine, or ISO-DE. Its entire job is to predict the exact 3D shape of that hidden pocket and then actually generate the molecule, the key that fits into it.
And the performance stats on this thing were wild. Yeah. The report had a direct comparison to AlphaFold 3.
Yes. They used a really tough benchmark called runs and poses. It tests how accurately the AI predicts how a drug binds to a target. On the absolute hardest category, targets that look nothing like the original training data, AlphaFold 3 had a success rate of about 23.3%.
And the execution agent?
50%.
Wow.
It literally more than doubled the success rate on the hardest problems in all of structural biology.
That is a massive jump. What is ISO-DE doing that AlphaFold isn't?
It aligns with real-world physics. It uses a system called packflow. See, an AI can easily draw a 3D shape that looks like it fits together perfectly on a screen. But in the real world, atoms have actual forces.
Right, they repel each other, they attract each other.
Exactly. If you jam a drug molecule into a pocket where the electron clouds of the atoms overlap too much, the real world just says no.
The molecule would just bounce right out.
Or it would tear the protein apart. Packflow mathematically ensures that the prediction sits in what's called a low-energy basin. It guarantees that the drug fits the shape and that the laws of physics would actually allow it to stay there naturally.
Now, speaking of staying there, I want to bring up something that felt super important in the notes, but I didn't fully grasp it at first. Entropy. The report mentioned this training data called the PB database, and something about an entropic penalty.
Ah, yes. Entropy. This is the forgotten child of drug discovery, and it is so critical.
I mean, I know entropy roughly means disorder, right?
Yeah.
The universe likes things getting messy.
Right, the second law of thermodynamics. The universe tends toward disorder. It likes chaos, and it loves freedom of movement. Now, imagine a drug molecule floating freely in your bloodstream. It's tumbling, it's spinning, it's vibrating. It has total freedom. High entropy.
Okay, tracking.
Now, imagine that same drug binding tightly into a cryptic pocket on a protein. Suddenly, it is stuck. It's locked in place. It can't tumble. It can't spin.
It lost all its freedom.
And the universe hates that. That loss of freedom is an actual energy cost. Scientists call it the entropic penalty, or delta S. Even if the shape of the drug fits the pocket absolutely perfectly, if the thermodynamic cost of locking it down is too high, the drug will just pop right back off into the bloodstream.
So it's kind of like trying to seat a really hyperactive toddler in a rigid wooden chair. Even if the chair is exactly the right size for the kid, the toddler has so much energy they just won't stay in it.
That is the perfect analogy. Most AI models totally ignore the toddler's energy. They just measure the chair size.
But the execution agent doesn't.
Right. Using the PPI database, which has hard thermodynamic data, the agent actually calculates the energetic cost of sitting the toddler down. Through its generative model, which they call O-design, it models the flexibility of both the protein and the drug simultaneously. It ensures the chemical attraction is strong enough to pay that entropic penalty.
That is fascinating. It's accounting for the physical personality of the molecules, not just their geometry.
Precisely. Co-designing the lock and the key at the same time.
Okay, so we've cracked the safe. We have a drug that physically and thermodynamically works. But wait, we are still dealing with AI here. And as anyone who has used the standard chatbot knows, AI can be a very confident liar. How did the scientists know this digital crew isn't just making the whole thing up?
Enter the fourth and final member of the team, Layer 4, the Auditor agent.
The guy in the van, or I guess the gatekeeper.
The necessary buzzkill. The Auditor's sole job is to ruthlessly fact-check the other agents. It uses a technology called GraphMERT, which relies heavily on knowledge graphs.
Knowledge graphs, that's like a giant web of facts, right?
Yes, it breaks all of known biological literature down into triples. Head, relation, tail, like protein A interacts with molecule B, or kinase C is found in the liver.
So if his execution agent says, hey, I found a great pocket on the knee of this protein.
The Auditor checks the biological knowledge graph and says proteins do not have knees. Try again.
Does it really catch that many mistakes in practice?
Oh, absolutely. They ran a comparison study on diabetes targets, and they found that standard large language models had a factuality score of only about 40%.
40%. That's failing.
It's completely failing. They were hallucinating biological pathways that simply did not exist.
That.
But the Auditor agent using GraphMERT brought that accuracy up to nearly 70%.
That's a huge safety net.
It is. And it doesn't even just rely on its internal memory graph. It uses something called the cryptic framework.
Oh, this was the part that actually creeped me out a little bit. The AI can use external tools.
It does. If the auditor is unsure about, say, a specific chemical bond length, it doesn't guess. It can literally pause, open a Python interpreter, and run a mathematical calculation code itself. Or it can ping a search engine to verify a fact from a recent paper.
It's Googling its own homework.
It really is. And just by double-checking its work using those external tools, it reduces reasoning errors by over 30%. It creates a self-correcting ecosystem.
Okay, before we get to the really crazy futuristic stuff, I need to understand how these agents are actually talking to each other. Because proteins aren't text. You can't just type the letter A into a terminal and describe a 3D shape. How does the AI read the physical geometry of a protein?
This is a massive breakthrough called GEOBPE, geometric byte pair encoding.
Okay, English, please.
Well, you know how standard AI models read text? They don't read letter by letter. They break words into tokens. Like the word apple might be one token, the suffixing might be another.
Right, chunks of text.
GEOBPE does the exact same thing, but for 3D geometry. It breaks the sweeping backbone of a protein into geometric sentences. A specific twist, a hay-pin turn, a beta sheet. These physical motifs become the actual words in the AI's language.
So it's translating a physical structural shape into a digital language it can process.
Yes, and it's incredibly efficient. By tokenizing the structure this way, it compresses the data size by 10 times, but it keeps all the important meaning. It aligns perfectly with known functional families, like the CS database. It allows the AI to see the functional logic of the protein, not just a massive, confusing cloud of atoms.
It's seeing the forest, not just the trees.
Exactly.
All right, so we have the multi-agent crew, we have the geometric language, we have the rigorous physics. But the report ends with something that, honestly, I had to read this section 3 times. It sounds way less like biology and way more like the technological singularity. We need to talk about the Darwin Gödel Machine.
I was wondering when we get to this. This is definitely the climax of the paper.
It sounds like a superweapon from a movie. What exactly is it?
It is the mechanism for recursive self-evolution. We aren't just talking about an AI that learns from new data anymore. We are talking about an AI architecture that literally rewrites its own reasoning code.
Wait, pause. It modifies its own source code?
Oh.
While it's running?
Yes. It's built on a concept called agent evolver, and it operates on 3 mechanisms. First, self-questioning. It actively invents its own training tasks to push its own boundaries. Second, self-navigating. It meticulously remembers its past attempts so it never repeats a dead-end mistake.
And the third?
Self-attributing. When it finally succeeds at finding a pocket, it figures out exactly which line of logic or which specific action led to that success, and then, and this is where the Darwin Gödel part comes in, it preserves that logic. It maintains an archive of agents, like a digital fossil record.
So it's survival of the fittest, but for software logic.
It is pure Darwinian evolution applied to AI. It keeps these stepping stone agents, versions of itself that maybe weren't perfect, but they had one brilliant new idea. It combines them, it mutates them, it constantly evolves new strategies that the original human programmers never even thought to code into it.
That is incredibly intense. So we are really moving away from the old days of virtual screening, which is basically just checking a massive catalog of existing drugs against a target.
Right, trial and error.
And we are moving toward predictive world modeling.
That is the ultimate paradigm shift here. We are no longer just looking up answers in a chemical dictionary. We are building a dynamic digital world that accurately simulates the complex physics of biology. And then we are releasing a self-improving, self-evolving species of AI agents into that world to hunt down the cures for us.
It really feels like we are standing on the precipice of a totally new era in medicine. We're going from throwing spaghetti at the wall to having a coordinated team of sniper robots finding hidden targets that we didn't even know existed.
And when you consider that the undruggable proteome includes the primary drivers of things like pancreatic cancer, Alzheimer's, severe autoimmune disorders, this HNS-MAS suite isn't just cool computer science. It is quite literally a lifeline for millions of people.
It's solving the unsolvable puzzle by entirely changing how we look at the pieces.
And by changing who, or rather what, is doing the looking.
I want to leave you all with a thought based on that Darwin Gödel Machine concept. If this AI system can recursively rewrite its own code to become a better scientist, if it can evolve its own logic beyond its initial programming, are we fast approaching a point where the AI discovers a biological cure or a mechanism of disease that is 100% correct and effective, but is fundamentally too complex for human scientists to even understand?
That is the real philosophical question here. We might very soon be curing terminal diseases using molecular mechanisms that we as humans can no longer explain. The agent will understand it perfectly, but we might just have to trust the code.
A completely fascinating, slightly terrifying, but mostly incredibly hopeful place to end today. Thank you for diving deep with us.
Always a pleasure to be here.
We will see you all next time.